Inviting our third and last speaker in the neuroscience session. This is welcome with me, Dr. Shahab Asgharzadeh. He's an associate professor of pediatrics at the Keck School of Medicine at the University of Southern California. He serves as the director of the Neuroblastoma Basic and Translational Program at CHLA, along with being also a director of the Spatial Biology and Genomic Core at the CHLA. Dr. Asgharzadeh earned his bachelor degree in biomedical engineering from Northwestern University and a medical degree from the University of Illinois. He completed his training in pediatrics and hematology oncology at the Children's Hospital, Los Angeles, prior to joining the faculty of USC in 2005. His research focuses on understanding the tumor microenvironment's role in peripheral and central nervous system tumors, particularly high-risk neuroblastomas and medulloblastomas. And today he speaks on exploring the neuroblastoma tumor microenvironment and spatial biology perspective.
Thank you very much. Thank you, Alex, for the invitation. So this is not exactly a neuroscience talk. It's neuroblastoma is a peripheral nervous system tumor. I'll talk a little bit about the developmental aspect of it as well, but it's really a childhood tumor. So I'll go over a little bit about the tumor microenvironment in this disease, tell you a little bit about the disease, talk about some of the single cell and spatial biology that we've done to look at the crosstalk between tumor-associated macrophages, TAMs, and cancer-associated fibroblast CAFs and these tumors. And I'll throw in a little bit of at the end of a CAR T cell design that we've looked at to combat this tumor microenvironment. There's a little bit of therapy at the end as well. So neuroblastoma is a tumor of children. It occurs majority in kids less than 11 years of age. It's derived from the neural crest cells, and cells that are destined to become the sympathetic ganglion cells that form your adrenal gland. These are nervous system just outside your spine. And people for many years thought that neurocrest gives rise to all of these cells, and we'll go through a little bit about that is not exactly the current viewpoint and why that matters in neuroblastoma. But the general thinking is, we get these tumors that are solid right above your kidneys. The kids present with metastasis to the bone and the bone marrow. And we have a specific radiographic dye, radionuclide dye called MIBG, that when you give it to the patient, you can actually see where the tumor has gone, and this child has had tumor go through lots of different organs, including bone and bone marrow. It's divided into different categories. A very interesting disease. It's one of the few human cancers that you could have a metastatic disease in a child at less than 1 year of age, and we do nothing. We don't give chemotherapy, and the tumor goes away by itself. So it regresses. It's only in the infants that occurs. We don't exactly know why. But if you're older than 18 months of age and you get that same metastatic disease, the genomics changes a little bit, but those child has a survival of only about 50%. So those patients that have metastatic disease, typically older, or a particular one that have a MYC amplification, so oncogene, have a really poor outcome. So on the bottom, I showed you that's an MRI of a liver. Those are lesions of the tumor. That's the special stage 4S. So these kids come in with huge livers at sometimes right after birth, and we give no chemotherapy, and the tumor completely goes away within a year or so. But for a majority of the other kids, the survival is 50%, and that's what we are focusing on. So it turns out the age also is very interesting in neuroblastoma for not just that special case that I mentioned, but cases that don't have this amplification of that oncogene MYC, and kids less than 18 months of age get very minimal chemotherapy, and they survive. They are full-blown metastatic. They're not just very specific to the liver. But kids who are older than 18 months do really poorly. So many years ago, when I was a fellow, I was interested in figuring out why do kids who are older than 18 months of diagnosis get this really bad tumor. And we did some gene expression analysis with gene arrays back then and RT-PCR. And what turned out, and this is a heat map basically showing from age, it's sorted by age, and you appreciate that the kids that are by diagnosis at age and older than 18 months have more of a red. And these are a lot of genes that relate to the immune cells. So particularly actually the macrophages, and the black ones are the patients, the two blacks are the patients who've died with the disease, and no black means they survived. So when we looked at this a little further, we found out that the kids who are older with this particular metastatic disease, these all children have metastatic disease here. Uh, the tumors are filled with macrophages. And after you could took basically a signature of some tumor features as well as this inflammation signature, which was related to macrophages, the ones that had this inflammation signature did really poorly. And there was a direct correlation from our data from gene expression, the target data set, comparing TGF beta and the CD163, which is a marker of macrophages. So several years later, we looked at just the serial immunohistochemistry, and this is a collaboration with Yves de Klerk. We looked at basically several markers and what series of basically low and high risk patients at diagnosis. What we noticed that the primary cells that are in these tumors are consist of macrophages, the green ones, and CAFs, the purple ones. So there's very few T cells or other cells in there. And there seemed to be a co-localization between macrophages and CAFs in these tumors. This led us to go a little bit more in depth and figure out, okay, if the kids at diagnosis have this microenvironment, they get tons of chemotherapy, they get a bone marrow transplant, they get radiation therapy. When the tumors come back, do their tumor microenvironments the same as diagnosis? Has it changed? Are there any even immune cells in there anymore? Because when they relapse, they have a really extremely poor survival. So once you relapse or you're refractory to the chemotherapy initially, you have a really poor outcome. So with this, we set up a clinical trial. Well, it's actually a biological trial, really, looking at the tumor specimens relapse from 14 large children's hospitals across the United States. I chaired this trial, and we were trying to both do a genomic analysis as well as immune environments with just markers of macrophages and PD-L1, send a report back to the families and the physicians to help these children with deciding on what type of therapy to do next. In the process, we actually took a lot of these tumors that were both at diagnosis, they were at relapse and refractory, and we put them through an image mass cytometry using the Hyperion system, and we developed a 38 marker TME panel. And again, what we noticed, which was really interesting, is that we still have this predominance of TAMs and CAFs. There seemed to be, again, on the top right, the marker works, but the top panel is on the left is diagnosis and is refractory. Refractories are tumors that do not respond to chemotherapy at all. These tumors just blow through chemotherapy. And the one at the bottom is a relapsed. But there's not a huge much difference. So the TME in a relapse and refractory is really similar. So your macrophages don't change, your CAFs don't change. So what is exactly that's changing here? And this led us to more of a wet bench type of work. And we've looked at the animal models of neuroblastoma. We have a very faithful recapitulation of the tumors growing in these mice that are driven by a tag oncogene. And we could take these tumors at different age times and realize that once they get to be a certain size, that macrophages infiltrate these tumors again. That's at the bottom. And then the levels of TGF beta and CCL2 is a marker that the tumor cells secrete. They're actually an invitation for monocytes to come into the tumor. They also go up around the time that they're getting very larger. And when you co-culture tumor cells with macrophages, the mouse, the tumors grow faster in the ones that have the macrophages. There's a lot of papers that we wrote looking at this association. We were really focused on the tumor-associated macrophages, but the bottom line is that the tumors help the macrophages help the tumors grow through upregulation of c-Myc and phospho-STAT3, and the tumors then secrete cytokines and chemotactic factors that bring in monocytes and then change them into macrophages. And there's a lot of other papers that we did that showed some of the chemoresistance aspects of these things through a mirror and exosome generation as well. But we got interested in this TAM-CAF study with another collaborator, Yves de Klerk, and asked the question, if you put CAFs in a tissue culture and throw monocytes on them, what happens to the monocytes? And this graph basically shows on the left if you have just the monocytes in culture, after 4 or 5 days, they start dying. But if you actually have some mesenchymal stromal cells, which are the precursors to CAFs, in the culture, and even low ratios, the monocytes remain alive. So they're actually keeping monocytes alive. And we've done this with CAFs from the patients directly, from the CAFs that are derived from bone marrows. Regardless, there's this CAFs are basically protecting monocytes from dying. So that's the relationship between CAFs and monocytes. And when we put this in animals, again, all the way on the right-hand side, the red signal means the tumor is growing faster. Those are the CHLA-255, which is a tumor cell line, co-cultured into the kidney with both macrophages and MSCs. And those are the ones that grow the best in the animal. So this is, again, a quick synopsis of some of the effects that we saw. When you co-culture all of these things in culture, we see an increase in TGF-beta that's a lot more pronounced than either by tumor cells alone, with a co-culture with monocytes, or with the CAFs. What happens in this triple culture, the NK cells stop working. And when you give gallosertib, which is a blocker of TGF-beta, that increases, again, the NK cytotoxicity. And we also see that this collaboration, the three-way collaboration, also increases the drug resistance to doxorubicin. So these talk between the macrophages and CAFs is really significant, both in terms of bringing in tumor cell immune cells, as well as secreting immunosuppressive cytokines, leading to immunosuppression and drug resistance. So a postdoc in my lab, when we were doing these experiments, noted that, like, when we actually do this culture, so we do this in a transwell culture, when you have the tumor cells at the bottom exposed to both monocytes and MSCs or CAFs on top, when you replate them, they actually look very different right after you replate them. They have this elongation of cells versus the control ones are very round. And with some morphometric analysis and proved this to be the case, they have a very different shape than the control cells. This is just the tumor cells by themselves when are conditioned in these media. This is where I have to tell you a little bit about some of the aspects in the literature that I have found. We've known for many years that neuroblastoma cells in culture have these two different phenotypes. One's called a mesenchymal phenotype, and one's called the adrenergic phenotype. You can think about it similar to adult cancers if you're familiar with the epithelial to mesenchymal transition. It's happening in these neuroblastomas as well, where there are certain markers of the tumors which we've known that should be true identifiers of neuroblastomas, such as FOX2B. They get downregulated. There's an increase in SOX9 and fibronectin. And the cell lines that we have from patients, it can be grown in isogenic cell lines that have either a mesenchymal phenotype or an adrenergic phenotype. And our cells in the co-culture with the TAMs and CAFs after 3 days, really look like the mesenchymal phenotype in terms of their just morphometric and shape. So I'm going to give you guys a little bit just a background as well. The neurocrest cells also turns out not to be the main cells that give rise to neuroblastomas. There's these precursors, there's this hub of multipotent cells during the neurocrest development, because there's not enough neurocrest cells to develop all the organs that we think develop. There's hubs that are formed that are called Schwann cell precursors that give rise to all the typical lineage of organs and tissues that we think about. Single cell analysis also looks at when we look at the fetal adrenal gland, which is derived from these Schwann cell precursors, have these developmental origin of Schwann cell precursors going into neuroblastic chromaffin cells. This is basically the organ that gives you epinephrine, flight and fright response. And the Schwann cell precursors are really the sort of the progenitor cells in this fetal adrenal gland, and they're the one that would most likely look like the mesenchymal phenotype in neuroblastomas, at least in the cell line. So we had this method, and we saw that there's a hypothesis was that when we have these TAMs and CAFs, we could actually lineage change the neuroblastomas from this neuroadrenergic to a mesenchymal phenotype. People have not seen this in human tissues, but they have seen it in cell lines. So the first thing we actually looked at single cell analysis of the cell lines exposed to the TAMs and CAFs in culture. And we did a single cell analysis. On the bottom is the clustering overlaying basically both the two cell lines, one at the top and one at the bottom. They're either treated with inotransil with TAMs and CAFs, we call TME, or not. And for each individual clusters that map on top of each other, we ask the question, are they enriched for these mesenchymal signatures or adrenergic signatures? And with the co-culture, almost all of the clusters had an increase in the mesenchymal signature. And this was actually in pathway analysis, looked like there was a mesenchymal. Epithelial mesenchymal transition pathway came up, TGF beta receptor signaling came up, and some Notch signaling. And when we do the protein analysis, we actually confirm that these cells are actually becoming, at least a subset of them, into a mesenchymal phenotype. So the TME is really, even though there may not be that much of them, it's less than 5% of the cells in the tumor in the child, there may be co-localizing with these mesenchymal cells that are resistant to therapy, both in terms of immunotherapy as well as drug resistance. And we also then superimposed our data on top of this fetal adrenal map, and again showed that the neuroblastomas, the highest differentially expressed genes in the TME co-culture neuroblastomas, again light up in the SCP, so the Schwann cell precursors, versus the control ones are all more towards the more differentiated and cycling neuroblasts. So we went back to our NANS precision trial that we had done, where we had characterized all of these tumors by genomic status and diagnosis refractory, and did a Visium analysis on a subset of these. And basically, to the standard clustering, the top part is the image of the tissue, the bottom what's a UMAP of the same tissue, and did a gene set analysis for these adrenergic and mesenchymal signature for each spot. Now, we assume the spot is a cell here, but we know it's not. It's more than about 10 cells or so. But this gave us an idea of maybe where in the tumor there's this signal for mesenchymal signature coming from. And it turned out the bottom of this particular tumor was more mesenchymal, where the top was mostly adrenergic. And you can see that on the UMAP as well, where there's a stronger signature of either mesenchymal or adrenergic. Then we went ahead and actually did the same thing. Left one is the same. We did it for CAF signature and a monocyte and TAM signature. Again, I hope you appreciate they're also at the bottom of this tumor and highlighting the same UMAP area. So when you superimpose these things, the TAM and the CAF signatures occupy a certain space in the UMAP. The mesenchymal signatures occupies one part of the TAMs and CAFs that are completely blended in the yellow spot, versus there's no overlap between the TAM CAF signature and the adrenergic signature. And if you take every spot and give it a signature score for TAM CAF or mesenchymal adrenergic and just plot it as a correlation map, you'll see that there's a really strong positive correlation of the higher the TAM and the CAF score there is, the higher the mesenchymal score is, and conversely, lower the adrenergic score. So we did this for all of the 15, and we got the same results for all of the 15 tumors that we had. So this is basically just a correlation, negative or positive, and the CAF versus mesenchymal or TAM versus mesenchymal on the right-hand side, the TAM CAF correlating with the mesenchymal signature. They're all positive correlation. There was also positive correlation between TGF beta and the mesenchymal phenotype in these tumors. So all of the tumors, all of the spots we analyzed showed a similar pattern. But going back to this is again still as a 10 cell. So what we really have to do, and the question that people might ask is, how do you know there's tumors in those spots that you have a mesenchymal signature? And that's a difficult thing to do in Visium. And we're trying to look at copy number using InferCMV or copycat to try to estimate, or other tools try to estimate the tumor cells in that context. But we're also turned over to the SeqFish technology in collaboration with Long Kai at Caltech. And you guys know it's similar to a SeqFish that people have probably already known, but we could truly look at the single cell resolution and count about 500 genes per cell in these specimens. And this is data generated by Mikhail Polonsky, who will be talking, I think, in this session as well. And this is data still cooking, because we're looking at both high risk and low risk tumors, but we have a much better resolution now of the different immune cells. And on the right-hand side, some of the low risk are filled with a lot of other immune cells, including CD4s and CD8. So there's no longer just TAMs and CAFs, versus the high risk on the left, the three of them, still have a good number of fibroblasts and TAMs. So this is the next step for us to really hone in on the correlation between the TAM and CAFs and the nearby cells that may be mesenchymal. So I'll switch gears a little bit, just tell you guys a little bit about our therapeutic program in my lab. So we were interested in this TGF-beta story, and we know that the CAR T cells work really well in leukemias, but because of TGF-beta, whenever we made tumor cells that were truly solid tumors in mice and gave them the same CAR T cells that may work in a metastatic model or like in a tissue culture, these would not work. And we suspected this is because of the TGF-beta role in these neuroblastomas and many other cancers. So this is my cartoon for a very simple modification. You have a T cell, you have a modified T cell, we call CAR T cells. They kill leukemias perfectly. TGF-beta is the poison. What we created the portal that takes basically TGF-beta poison and converts it to a particular energy source for T cells that's not very well known, but it's basically toll-like receptor signaling, which is a very primitive immune response in many immune cells. So this is basically what we did. TGF-beta receptor is as a monomer typically on cells, and when TGF-beta binds, it becomes a dimer. So that was our first idea that we could actually combine this to actually create a signaling. So we combined the TGF-beta receptor 2 with a toll-like receptor activation domain that activates phospho-IRAC. I'm not going to go into a lot of details, but what this does is actually really powers up the T cells to be able to penetrate into the tumors. So this is basically a liver section of tumor cells. When we give untransfused T cells, the brown is a nuclear stain of FOX2B for neuroblastomas. A CAR T cell, this is direct against the antigen in neuroblastoma called B7H3. The CAR T cells go around the tumor, but don't infiltrate it. When we have the CTSR, they could actually obliterate these nodules in these liver tumors. This results in a significant increase in survival of the mice. As you can see on the right-hand side, either B7H3 alone or CTSR alone, all the mice die. And this works both as a solid organ where we inject the tumors into the kidney of the mice on the left-hand side, or when we inject the tumors IV into the mice, they still survive. Some of the mice that you see die with X's died from graft versus host disease, which is typically seen when you inject human T cells in NSG mice. This works also in Ewing sarcoma, which is another bone and muscle cancer in children and young adolescents and young adults, and also in pancreatic tumors. So this is a very exciting finding. We've actually been able to obtain a CIRM grant for this, so hope to start a clinical trial in a couple of years. And we also were fortunate enough, I think, to get a fairly good score on the HCAN, so we'll join Ken for doing some of this more work on the solid tumors in the upcoming years with Mikhail and Long. But the conclusion basically is the tumor microenvironment, I think not just only neuroblastoma, but many solid tumors are really important. Even though there may be very few percentage of the cells, those are the cells that may help the cells become more dedifferentiated, that allow them to become metastatic, set up shop, become more adrenergic or epithelial, and do their work. And there are actually, I didn't talk about it, there's a lot of small molecule work that we're doing as well to block that interaction. And CAR T cell therapy, I think, is a really promising way, if we could understand the TME and use it to our advantage, to create these novel cell therapies that could bust these TMEs. And with that, I thank my lab, my collaborator, Bob Akwilimi, who did a lot of the CAR T cell work, and Yves de Klerk, a lot of the TAM and the CAF work, and Mikhail and Long, we're doing the work with the spatial transcriptomics and spatial biology and genomics core that I lead with members of the core that are here that do a lot of the work for this study. And thanks to the patients and families who participate in our studies, and I'm happy to answer questions.
Wonderful, fascinating. And thank you for staying the time as well. Any questions? I have maybe one, please. one question. Something that caught my eye was Mir 155 microRNA. 155 is known to be changed in microglia in Alzheimer's disease in the brain. So it's highly associated with the plaque, with the lesion site, with the heightened inflammation and detrimental effect. And it's thought that if you inhibit it, it could have some positive effects on the outcome. What is your thought? Because I saw this was one of your markers.
It causes cisplatin resistance in neuroblastomas. So it's actually a marker for it's for drug resistance in neuroblastoma. So this is through the TAMS and correlation with macrophages. So work we did with Muller-Fabry many years ago, where he actually showed that these can actually bind to the exosomes and can bind to toll-like receptors and get uptake into the cells and cause chemotherapy resistance. It's into other things.
That's
Correct. Correct.
Yes.
That I don't remember, to be honest with you. It was 10 years ago. Yeah. So
Questions?
thank you.
Yep, you do.
Curious about the tumor diet. You mentioned that you see an increase in T-cell activation. Is that the number only, or is it that given to
In the low risk tumors, the spatial, yeah. So T cells are known to be in the younger patients, but not in the older patients. And it's actually quite interesting because MHC class 1 is downregulated in both low risk and high risk tumors. But it's more of a favorable prognosis as in like many other cancers as well, if you have higher T cells, but not necessarily all the time. We've actually done many years ago, they've done clinical trials where they actually give interferon gamma to patients, and that increases your MHC class 1 and brings a lot of T cells into the tumor, but no clinical benefit. So they still are not able to overcome whatever the tumor microenvironment barrier is as well.
Did they analyze patients? Helper T cell, suppressor T cell, different toxic T cell.
Yeah, Stephen Grupp is the same guy who did the CD19 CAR T cells, and that was 15 years ago. I don't think he actually analyzed those at the time either. So we don't know exactly. But I think even if you give interferon gamma and the T cells come in, there's still IL-10, TGF beta. There's a lot of immunosuppressive cytokines that you may be able to get them there, but they're not going to function.